I am Pier Palamara. I am based at the University of Oxford in the UK. It's been great to be here, to see lots of people again in person after a while and to meet lots of new people. And so today I'm going to tell you about this work. It's a pretty long title. It's the title of the paper we put out recently. But essentially, these are the key ingredients of the talk. I'll tell you a lot about genealogies and actually ancestral recombination graphs. It's just a technical way of referring to a specific type of genealogies that I'll tell you about, and ways of using this genealogy, inferring them, and using them to analyze things like complex traits, so like things like GWAS. And a bit more in detail, this is the outline. So I'm going to tell you about modeling of genealogies, what I mean by genealogy, and the kind of the stochastic processes that we use also to do inference later on. Then I'll tell you about algorithms that we've been developing called ASMC and an extension called the ASMC class, and our needle, two algorithms that we developed. And then finally, ways to analyze these inferred genealogies for things like GWAS, so complex trait analysis. Okay, so starting from modeling and background. So when I say genealogy, you probably think about something like this. So a pedigree, where you have a pair of ancestors, a mother, a father, and two offspring. And so these are diploid organisms, so they have two haploid genomes each. And because of recombination, you're going to get the shuffling of genetic materials. In the next generation, the offspring are going to get some shuffling of the chromosomes of the parents. And this shuffling is random, and it's different each time there's a child. So what happens if you have more generations? So if you have a pedigree that is a bit deeper, you have fourth-degree cousins in this case, so they share ancestors 5 generations ago. And it may happen, it doesn't have to happen, but it may happen that you share some regions of the genome, which is called identical by descent, which is coming from the same ancestor here. So this genome region for both cousins comes from this genome at the top. And so the question is, what happens here in the white regions of the chromosome? You still have some ancestor. So if you go deep enough in time, you're always going to find a common ancestor. And so I guess the point I'm making here is that a tree here is not enough to visualize, to represent ancestry when it comes to chromosomes. Even for a pair of chromosomes, you need something more complex, like a graph. So here we have a loop, so you need some kind of more complex graph, and that's because of recombination. So the structure that we want to use to represent genomes, including recombination, their ancestry and the connections, is called an ancestral recombination graph. And it has some properties. Importantly, also, there is some sort of distribution over which kind of graphs you expect to see in a population. And so these kind of distributions are the things that we try to use when we do inference from data of these kind of structures. So I thought that the easiest way to tell you both what kind of structures we're looking at and something about the distribution is to show you how we would simulate these graphs and also how we would simulate genomes. All right, so this is a simulator that we're trying to write. We have 3 chromosomes. This could be my 2 maternal paternal chromosome and somebody else's chromosome 1. And so we have time on the y-axis here, and we do 2 things. To simulate a realistic graph in a population or in a sample of 3 individuals, we would either coalesce genomes. That means that we take 2 nodes that we're tracking and we merge them. And we do this with a rate that depends on the number of pairs of nodes that we're tracking. Or we recombine them. A recombination looks like this. So you take a node and you split it at some recombination breakpoint. And so this increases the number of nodes that we're tracking. The amount of genetic material doesn't change here because I'm just sending some material here in gray and some here. And this happens at a rate that depends on how much material I'm tracking. And so this is this exponential waiting times for these kind of events. There are some details that don't really matter. And the intuition here is that, for instance, for coalescence, if you're looking for ancestors, if you're sampling ancestors back in time for several or for a pair of individuals, you have a small chance of picking the same ancestor, depending on the population size, at each generation. So you have a geometric waiting time or exponential in the limit. So there are some details of what the distributions look like, but I'm going to skip those. And there's a lot of literature. And the process that I'm using here to simulate is called the coalescing with recombination. So you can read more about that if you're interested. And here are some references. Okay, so if we keep running this, so we keep getting our sample args, this graph for the simulation. There's a coalescent event that happens here, so I take this node and I merge them. And now this amount of material has been merged, so it's colored in blue. And this happens again on the left. And finally, you get this node, which is the ancestor of everyone in this example. And so here we stop simulating. So we have a complete graph, a complete arg. So this, the arg has a number of interesting properties. So one of them is that if I slice it at a specific position, so if I only keep the nodes that overlap position, say P1, then what I get is a marginal tree. So the way if you stare at this for a while, maybe you see that what's happening here is when I go backwards in time here, I'm routed to the left because the gray material here is on the left, and then I go this way. This node is not really useful because it's just a unary node. I just traverse it without doing much. So if I slice the graph and I only keep the edges and the nodes at this position, I get this tree. And if I slice it on this side, I get this other tree. So I have these two marginal trees. So if I take this position, I have this one. Otherwise, I go right here and I have this tree. So this is a property that an arg, if you slice it at some position, you get a tree. And actually, there is a duality between a bunch of trees and a graph like this. It's just the same way of representing the same information. There are some philosophical points, but for this talk, it doesn't matter. These are equivalent representations. And if you want to simulate data, so if you want to get genomes, if you're writing your simulator, now you have a way of getting the graph. If you want to get the genomes as well, all you do is you sprinkle mutations on the graph using a Poisson process for the same reason that you have exponential waiting times for the other events. You have the same type of waiting times for mutations. And so you throw mutations on the graph, and you get mutations on the marginal trees, and the descendants of the branches that are hit by mutation get the mutation. So this is a way of writing your own simulator, and it's also a way of getting a sense of what kind of distributions and processes that are behind this kind of objects that we also use for inference. And something that is also going to be useful for this talk is that sometimes or often I'm going to focus on the arg of two individuals, so a pairwise arg. And the way you get a pairwise arg is, in this example, you just remove these nodes. We only care about two samples. And so you get this kind of structure. And now the marginal trees at the two positions that I had before just look like this. You have just a node at the top, and you're reaching that ancestor somehow. And the thing is that these trees extend for some amount of genome. The tree on the left extends between position 0 and wherever recombination happens. And the one on the right extends from recombination to end of chromosome. So another way of showing the same information is this, which I'm going to use later on as well. So this is just a way of showing you a pairwise arg, which represents the two haploid genomes at the bottom. These are the ancestors. And the height here is called also TMRCA, or time to most recent common ancestor. And so you get some kind of piecewise constant function along the genome that describes the arg for two samples. And again, if you want to simulate two genomes worth of ancestry and SNPs and genomes, once you have simulated this kind of graph, all you have to do is you add mutations with some kind of Poisson process. So you have at each block, for each segment that I have here, I just sprinkle mutations with Poisson random variable. The number of mutations is Poisson, and then where they land is just uniform on either lineage. And that's how I can add mutations. For each block, I can do this thing. Okay, so that's all I wanted to say about background, genealogies. And now what we want to do is to use this kind of modeling to infer. So you go from the genomes themselves. You want to figure out what the graph is underlying the population or the sample. And this is actually a fairly messy problem because if you go back actually and think about the number of combinations of graphs you can get out of this process is huge. And we're trying to do inference based on the mutations, which is a very small amount of information with respect to where the edges and the nodes are in the graph. And so there's a number of ways that we can think of doing this, but it's a fairly complex problem, which makes it fun. But besides being fun, I should also tell you why it's interesting, or which kind of things we're hoping to get. So why do we spend all this energy trying to get genealogies? And so I was saying that the genome is a product of the genealogy, essentially. So if you have the genomes that we can collect, they're essentially a projection of these processes and this graph that is behind them, which is a genealogical structure, or an arg. And the arg itself is sort of the parameters that that the effect which shape these graphs take, the distributions, are things like natural selection, or the demographic history of the population, or through selection, things like heritability and complex traits and so on, which are the things that we normally want to study. And so if you start from the genome, usually what you do is you get some kind of summary statistic like haplotype frequencies or allele frequencies. You kind of marginalize out the genealogy and try to get information about these things. So if you manage to get the genealogy, you're quite close to the source of what you're trying to study. So that's the reason essentially that we're trying to do this thing. Okay, and so I'm going to tell you about some algorithms and results, very briefly, just results, examples of results from these papers that we've been working on in the past few years. Okay, I'm going to start from, so now moving on to inference. I'm going to tell you about this ASMC algorithm, which sort of tries to get the genealogy, the arg for two haploid genomes, two chromosomes. And so this is the process where we left. So this is how you can simulate haploid genomes. The mutations that we find if I sequence someone, if I sequence myself, this could be my maternal and paternal chromosomes, are just a result of these kind of processes. And so I don't see the arg, I don't see the graph. That's what I want to infer. All I have is data here at the bottom. And so I'm going to describe something that is called PSMC. It's an algorithm that was developed by Lee and Durbin in 2011. It's a very cool algorithm. And PSMC stands for pairwise sequentially Markovian coalescence, and that's because it uses all these kind of formulations of these processes here at the bottom. But essentially what is, at a high level, this is quite simple. So what you do is you take these two haploid genomes, and then you compute the XOR. So for each position, you keep a 1 if they're different. So in this case, this chromosome has a mutation, this doesn't, so we have a 1. If they're identical, it could be homozygous, derived, or ancestral, we don't care, you have a 0 here. So we just compute the XOR of the two bit sets, the two strings that describe the chromosomes. We can throw away the genomes, we keep the XOR, and then we use an HMM, so a hidden Markov model, where we need some hidden states, which is what we're trying to infer. So the states here represent where the ancestor is along the genome. So remember, we're trying to infer a piecewise constant function of where the ancestors lie. And the observations are just the genome in this form. The hidden states are a discretized version of time. And so the emission is actually reasonably simple because I said this is a Poisson number of mutations. So this ends up looking like an exponential probability. So the emission is quite easy to write. The transition is a bit more messy. You have to write a few expressions about the probability of coalescing at each time point within each interval, recombining, and then coalescing somewhere else. But it's doable. It's actually quite fun. And eventually you get some kind of matrix that describes the transition probability. So that's the two main components that we need. We need an initial state distribution, which is computed similarly with a transition. And so you have this tool, PSMC, that allows you to do this calculation. So you get an HMM that outputs, given an input XOR of genomes, outputs a posterior probability of coalescence time, or TMRCA, or the pairwise arg, many ways of calling this, but it gives you ancestry, the time distance of the ancestors as a function of position along the genome. So this is a cartoonish view of this, but if you now do a simulation, a bit more realistic, so the red lines here are the true simulated ancestors, the time depth of the ancestors, and the heat map is the posterior that you get from the HMM. And you can see that it's tracking the red lines pretty well. Now I'm going to skip a lot of details, but the algorithm that I mentioned earlier is called ASMC, which is an extension of this approach. And the goal of this extension, which is something that I was working a few years ago, is to be able to work with SNP array data, so not only sequencing, but also SNP data. And the complication here is that you have a lot more sparsity, so you have to jump more far in the transition. And the emissions are not just mutations anymore, because you have to think about the frequencies, because when you have SNP data, you normally just keep the high frequency mutations. And the other ingredient here is that we want this to be quite fast, because we want it to be able to be applied to large data sets. So when you do forward-backward calculations in an HMM, you normally have to do a matrix vector multiplication a bunch of times, where the matrix is a transition matrix, and the vector is some state vector for the probabilities. And that would be normally quadratic in the size of the vector, which is the number of hidden states here. And you can actually leverage some symmetries in the transition matrix to make this become linear. So at the end of the day, you get something that is faster and can work on SNP array data. And the reason why we want to work with SNP data is that sometimes we have data sets for which we haven't collected sequencing. We have a lot of SNP data, and that's interesting to be able to do this kind of stuff. Okay, so that's the first ingredient, is this sort of, we have some tool that gives us, given 2 genomes of sequencing or array data, these kind of pairwise args. And this is just another simulation. This is on the top is something like PSMC using sequencing. You get a good correlation between TMRCAs and inferred TMRCAs. In SNP data, you get much fewer observations and a bit sparser data, so you expect it to be doing worse, but it's still reasonably calibrated. Particularly here at the bottom, if you're looking for haplotypes, they're reasonably accurate. Okay, so I'll briefly show you a couple of examples of what we can do with this kind of information, or other methods you can build on top of this tool. And I said I would focus on, the title is about complex race, so we'll get here eventually, and I'll spend a bit more time, but when I get there, I'm going to show you a couple of things about demographies, demographic history, and also natural selection later on. So with this tool, we can do a few things. One thing that you might be interested in is if you have a big data set, you have a bunch of genomes, you want to figure out who's related to who, you want to figure out if people tend to have close ancestors in the past, say, 1,000 years or something like that. And that's something that you won't have a pedigree for. People are not aware of this kind of relationships, and the genome kind of has that information. So one thing you could do is you could say everyone below a time threshold is IBD, identical by descent, and that's kind of looking for ancestors that are shared for pairs of individuals. They're likely not aware of that, but individuals which are cousin of some degree in a data set. And so if you do this in the UK, so this is now data for about 1 million haploid genomes in the UK Biobank, so SNP data. We run this approach. I'm skipping some details, but we ran something very close to this approach, and we detected a few hundreds of billions of segments. These are sort of ancestors. So this is in the past 1,500 years. These are ancestors that these pairs of individuals are not aware of having. And what we have here is a matrix where you have in position I and J the number of ancestors or segments. Actually, an ancestor can transmit multiple segments, not very common, but depending on what threshold you use here. But a modulo some details, this is sort of representing how many ancestors individuals I and J have in a big matrix. And this is sorted more or less by north to south. So you have UK coordinates here, and these are actually averages for postcode. So for each postcode in the UK, we have position I and J, average number of ancestors across individuals. And so a few patterns that come out here is that the diagonal is very large compared to the rest, and that's because people tend to have distant cousins close to where they live, which makes sense. The north has a lot more sharing, so Scotland and north of England have more shared ancestors because the effective population size is smaller, so people tend to have closer relatives. And this cross here is London, which is cosmopolitan, so you have ancestry from all over the country, and this locality doesn't really apply. And so if you have this big matrix, it's a sparse matrix, so people tend to not have many ancestors. You can take it and do hierarchical clustering or other analysis on top of that. And so what you get, depending on where you slice this dendrogram, so at the bottom here you have a little tree that represents the whole ancestry. Depending on where you slice this tree, you get clusters that are bigger or smaller. And so this is, say, certain heights. You get some clusters that tend to be localized in regions and large cities and so on. If you keep zooming in and you slice the tree a bit lower, you keep getting these extended families that live around villages. And actually, so IBD is very predictive of where you're born. You can predict birth coordinates using IBD with a reasonable accuracy. And actually, there is a website at this address. You can go and click around and play with different postcodes in the UK. And if you click, it's going to show you where the distant cousins of people living in the postcode live, and you can tune time as well to get some sense of how things change in time. Okay, so natural selection, another example. Eventually get into heritability-related things, but natural selection, another application you might want to do with genealogies. Very briefly, a sketch of the method is you take all these pairwise posterior decodings of where the ancestors live as a function of genomic position, and you have a bunch of pairs, and then you average these posterior matrices, and you get this thing in the middle. And this is X is genome, Y is time, and the heat map is how many ancestors they share. And you have this kind of burst of sharing of ancestry here in the middle, and that's because of selection. As I mentioned, selection affects the shape of the genealogy. So in this case, you have selection is pulling the genealogy somehow. So you have more ancestry in the recent time. This is actually lactase for UK Biobank samples. And you can see, as we know for these locusts, there is strong selection in the past few thousand years after the development of agriculture. You have this burst of ancestry here. Okay, just a quick example about selection as well, but moving on to going from pairwise ARGs to full ARG. So we have a tool that gives us the ARG for 2 genomes, we want to get the ARG for the whole sample. And so this is the kind of extensions that we're building to use the ASMC type analysis for a more complete genealogy. This is work that we've been doing, lots of people in the group, particularly Brian, who defended his thesis on Monday on this project, actually. And there's a preprint has been up for a few months. If you want more details, and I'll just tell you very high level what the work was. Okay, so we have this tool, ASMC, that gives you this kind of graph, this kind of output. So some kind of piecewise constant function as a function of genome tells you the height of the ancestors. So this is a pairwise ARG. And what we can do is, this is reasonably fast because it's been optimized for speed. So you can do something like you do all the pairs in your sample. So you have n choose 2 pairs, you get n choose 2 pairwise ARGs, and you get something like this. And this information actually can be rearranged. So you have some big tensor where you have at every position in the genome, you have the TMRCA, they have the depth of the ancestor for individuals I and J stored in this matrix. It's the same data that I have here. It's just that here I have the height, the ancestry for each pair of individual. I can just arrange it in a matrix. So I have this kind of structure here. And now from this, if I want an ARG, remember there's a duality between trees and ARG, modulo some philosophical details. But what I can do is I can just run hierarchical clustering. I run UPGMA, complete linkage, and I get trees for each matrix. And so this is a good proxy for my ARG. And it's actually a reasonable way of getting your genealogies out of your pairwise ARGs. And the issue here is that this is a reasonable algorithm. That's well, we're going to keep it around as a baseline. And in general, it can be used and it gives interesting results in some cases. But it's quadratic, so it doesn't scale to very large data sets. So if you have n samples, you're going to have to work n square computation to get your trees. This step doesn't add complexity because the n square is already here. So getting the pairwise ARGs for n choose 2 samples is already quadratic. So this doesn't add much complexity, but overall n square is not too great. So instead, what we do is this thing called ARG needle. So we have another algorithm that threads one sample at a time into an ARG. This is called ARG needle is inspired by another algorithm called ArgWeaver that used a related strategy a few years ago. And so it's a kind of an inductive approach where you assume you have already built the ARG for n samples, and you can start for 2 samples. You have a pairwise ARG from ASMC. You have the ARG of n samples. I'm just going to show you the tree, but I mean the whole ARG, or this is just the tree at one position in the ARG if you slice the graph. But this thing happens for the entire genome. So if you have the ARG already for n samples, and you have a new sample you want to add, we do the following things. We assume we have already built something called a genotype hashing database. So if you haven't heard about this, you can look at these references. But essentially, it's something where we take the genome, we slice it into windows of little haplotypes, we throw them into hash maps. And then when you have your new sample, if you want to quickly find closest cousins candidates. So the goal of the first step out of 3 in the algorithm is to find a subset of all the people in the ARG that are closely related to the one that I'm trying to thread, to add to the ARG. So to do that, I have this hashing step where I want to quickly find a set of k closest cousins that are candidate, IBD, IBS. So they have long haplotypes that are identical, which means that they're likely going to be cousins at this position. So the second step is once I have this much smaller set of candidates to look at, I use ASMC to get the time depth as well for these samples. And then I take the minimum of these time depths, and now I have a refined estimate of which one is the closest cousin at this site for this sample. There might be more than one, but we only care about any of them. And so I get something called a threading instruction, which means that that's all I need to actually add this sample to the arg. So if I know which one is the closest sample and what height I have to attach this branch to, then I'm done. I attach that branch, and I have an arg now that contains n+1 samples at every site, and I can keep going to the next iteration. So this is the algorithm, and again, it relies on a few tricks, including this ASMC step in the second step. And then we did a bunch of evaluation, and actually this could be its own talk. There's a lot of ways of trying to figure out how the similarity between two args or trees. But so the arg is, it's something slightly different from trees, but it's sort of embedding a bunch of trees. So one classic metric of similarity between trees, and to some extent args as well, is this Robinson folds metric, where you have two trees. You're trying to estimate which fraction of the clades of the trees overlaps, essentially. This is actually just working on marginal trees, which is not great. So we designed another metric called total variation, arg total variation. So what we do is so the arg defines a distribution over bit sets. So if you have an arg, you can generate mutations from the arg. And so you can see the arg as a way of generating mutation, a distribution over the mutations that you can get. So if you have two of these objects, you can compare them as a distribution using total variation. And this accounts for branch lengths and other features of the arg. And there's details in supplementary notes of the paper. And then speed, we want this to be scalable. So the quadratic method ASMC is good for accuracy, but it's a bit slow. It scales quadratically. Argneedle is scaling well. It's not the fastest method. TS-infer is faster, but you have more accuracy with Argneedle, and this is scaling well as a function of sample size. So we can apply it to large data sets like the UK Biobank, for instance. Okay, so now moving on to using these genealogies to actually do analysis of complex traits, things like GWAS and other things, which we are calling genealogy-wide association. So we're now moving to this part of these applications. And so the idea, the general principle here is always the same in a way. So because we are able to do these things with SNP data, not just sequencing, we have access to more information if we get the genealogy. And so again, an example, this is it could be everyone in this room. It could be a number of chromosomes that we have available. And at the position in the genome, we know there's going to be some genealogy that describes the relationship of these samples. And we expect to find some mutations on this tree, or in this position of around this position of the genome, we expect to find mutations. We don't know what the mutations or the variants more generally are until we go and genotype or sequence these individuals. So let's say that we go and genotype them. So we have some SNP array data. And usually SNP array data is high frequency mutations that tend to be at the top of the tree. So we get some information, but not all of it. So we miss all these gray dots at the bottom. These are the rare variants that typically don't show up in SNP arrays. They can be in LD, but we don't see them directly, which is a pity because we want to do a bunch of things with these variants. We want to test them for association. We want to see whether they're shared and do other analysis. So a common solution to this is you go and sequence some people. So you have an imputation panel. You have a reference sequencing panel that you can use to impute the other samples. And this gives you access to more variation. But the problem here is, besides the fact that you have to sequence, the problem is that you won't see all the variation because all you see is what you have sequenced. If you haven't sequenced a variant, you can't impute it. So if you get the genealogy, the idea is that you don't see the mutations directly, but you get a much better sense of which ones can exist because there's a huge number of mutations, 2 to the n, that can exist for these samples. And this shrinks the ones that can actually exist substantially. And so you can sort of test them. You don't see them, but you can test a much smaller number of things and get evidence for things like association and so on. So that's the principle, and you don't need to sequence necessarily. If you have imputation, if you have sequencing, you can use it, but you don't have to sequence necessarily. So we want to use this kind of data. So we have the genealogy we infer from SNP data alone. So we get a sense of these orange dots here, and you have some phenotype like height or other things you're interested in. And we want to find association. And one way to do one very good way of doing association is to use mixed models, which you have heard about in the past few days. And so this framework allows you to do things like heritability estimation, polygenic prediction, and association as well. And it relies on this kinship or GRM matrix, which at position I and j, stores some kind of measure of genetic similarity of your samples, which lands here in this term. And you can imagine, we have seen something similar before. You can imagine that if you have an ARG, you can get a good estimate of this kind of entries, which is possibly a better estimate of than the one you would get with SNPs alone. And so this is actually something that we have played around with. And so if you go back to this kind of image, the number of mutations that we get along the genome is going to be some Poisson random variable given a certain depth of the ancestor. And this is true for every block. So the total number of mutations, which is the genetic similarity or dissimilarity of this pair of samples, is going to be some Poisson number of mutations with an expectation that completely depends on the height of these segments. And so if you have SNP data where you don't see some of these mutations here, if you hide some of them, not randomly, if you hide the ones that are usually rare, you would get some biased estimate of relatedness. And if you do this kind of trick, you get an unbiased estimator using the ARG itself. And so this is now simulation to show that this approach leads to interesting observations. So now this is SNP data in green, sequencing data in gray, and the ARG in brown. And so the ARG doesn't actually use any sequence. It doesn't look at the data, at the sequencing at all. This is the true ARG. But this is just to show that if you estimate heritability, as we know, you get a gap between SNP heritability and the true heritability you would get in sequencing. In prediction, you get a boost of prediction from using the SNPs, but you would get a bit more accuracy by using the sequencing. And association also, you would increase power if you used this kind of kinship estimates. And if you use the ARG, you get essentially the same results you would get from using the sequencing, which is an interesting observation. But anyway, more practically, one thing that we can actually do very easily, which is quite interesting, once you have done this inference, you have the ARG, and you have a good sense of where these dots could be, we can use the mixed model to do the association. So we can test each of these dots directly for whether they are associated with the phenotype. And so we can do what we call genealogy-wide association, which is very similar to a GWAS, but we just, instead of testing the mutations that we see, we test the mutations that are likely or possibly could be there. So the method is, it's reasonably, I guess, I have sort of described it, but I'm going to show you some results to show you what this might look like. So you start, so we inferred the ARG for the UK Biobank, so for these many samples, using only the SNP data, so we haven't used any sequencing data. But the UK Biobank is great because you have all sorts of other information to validate this analysis. So we have the SNPs, which we used to get the ARG. We have the ARG that we have inferred. I'm going to use these colors in the next slides to show results from each of the methods. And you have imputation, and at the time of this analysis was 65K haploid phase genomes from the HRC imputation panel and UK10K. And then we also have some exome sequencing data, so 50K, and then at some point 200K came out. And so what we can do is we can validate associations that we find using the exome sequencing for each of these methods. And so I'm grouping the results in two categories. The first is slightly less interesting, but it's very useful to visualize what's going on. And it's common variance and height. So association of height and common variance. And we know that these ones can be studied reasonably well with SNP data or imputation. But there could be things like structural variance, indels are not well called. So there's also some interesting things that can come out here. But then most interestingly, rare and ultra-rare variance in 6 quantitative traits. I'm going to show you that later. And first I'm going to start with this, so the common variance. And so here's a Manhattan plot. This is GWAS results for chromosome 8 genome position on the x-axis, minus log 10 p-value on the y-axis. These are the SNPs, the typed SNPs on the array for the samples we analyzed. And you have these 3 associations. These 3 SNPs are associated. When you do imputation, you go in sequence, reference panel, you impute, and you find this stuff. So you get a lot of more variants to test. You get these haplotypes, which are actually branches in the ARG. So the ARG contains some edges which subtend a bunch of samples that are tagging somehow carriers of the causal variant. So if you test these mutations, you get these plateaus and this kind of structure in GWAS hits, and that's because you're hitting branches of trees that extend for some amount of space in the genome. Now, if we go and build the ARG, essentially you're only using the green dots, so we're not sequencing any sample as it happened for the purple data. You infer the ARG and you test you do genealogy-wide association. Sorry, these are the haplotypes. So this is the kind of results you see. So you find the same haplotype structure that you would find if you went and imputed. You find the same type of plateaus and structure in a locus. And you sometimes also find these peaks, which are complementary, and they show you a bit more evidence, more structure at the locus, and they could maybe use it for fine mapping and other things. Okay, this is just to visualize what happened if you do this kind of analysis. And now moving on to the rare variants and ultra-rare variants, which are the things that we can't really impute very well because they're not sequenced. If they're not sequenced, we can't test them in the imputation framework. And so I'm skipping a bunch of details of filtering and validation, but essentially what we did is we took the exome data. For each association we found using either method, we looked for variants in the exome sequencing that would be tagging very strongly the association that we had found. And then we did a bunch of filtering to get independent signals. And so what you see here is for different phenotypes. In purple is validated signals that we found only using imputation. Orange is only using the ARG. And this is the intersection where you have found the same variant, exome variant, using both methods. So you can see there's a healthy overlap, but it's also complementarity. So for rare variants between 0.1% and 0.01% frequency, you find these kind of results. And if you move on to ultra-rare variants, where imputation is much less useful because we haven't sequenced variants at this frequency, for ultra-rare variants, you find a lot more stuff using the ARG than you find using imputation. There's some overlap, but in some cases, like mean platelet volume, it's only the ARG is finding stuff. And finally, last results slide is just to show you that we have, so this is the beta, so the effect estimated using either ARG or imputation, and this is the effect we estimate from the exome data, and you can see they're pretty strongly correlated, but the ARG finds a lot more sites. And if you look at properties of the exome variants that we have tagged in validation, then as you would expect, these are enriched for missense and loss of function variation. Okay, so just to summarize, genealogies are great for a number of things. It's algorithmically is an interesting and complex type of problem to approach, which makes it fun and interesting in many ways. And I talked about a few algorithms that we've been working on. And you can do analysis that relate to evolutionary history as well as complex traits, which is something that you can think of doing particularly if you have under-sequenced populations. So if you have a lot of sequencing data, then you have a lot of information already. But if you have SNP data for populations that are not being sequenced a lot, then this is something that you can think of using to get more information. And this is the work of many people. In particular, Brian has been leading a lot of the work for the needle algorithm. We have a preprint that's been there for a few months, if you're interested in more details. And we're recruiting postdocs, so if you're interested in this work, let's chat. And that's it. Thanks.